Firstly, I'd like to thank the ARM for allowing us this opportunity to give what I hope will be an exciting talk about a new therapy that we've been developing with other people. It's targeting what we think is a novel target, and we think it's got a really broad application for solid tumours. So like Oz has been talking about, we're targeting solid tumours. So in terms of Chimeric Therapeutics, I should just give you a background of what it is. It was established a number of years ago in 2015. It's a special purpose vehicle between a number of entities. It's ourselves, Cell and Gene Therapy Catapult. There's the University of Birmingham and the CRUK, Cancer Research UK, all of which are expert in either the development of these type of products and the regulatory requirements, the clinical trial requirements, or indeed finding the target and making it specific for what we want. It's been funded by ourselves and by CRUK. We've used our own core funding to fund this. And we actually don't use our own core funding very often unless we actually see something that's a particularly exciting therapy potential, and either that or it can break the barriers of how we might move forward the field. But in this case, we saw it as a particularly interesting target. We've got a comprehensive patent portfolio with it, and we are developing that still further. So as many of you know, there's a significant unmet need in the solid tumour field. It's worth hundreds of billions of pounds or dollars. CLEC14A, which is a target which I'll go on and explain about in the next few slides, has been shown to be present in multiple solid tumours. So there's a large potential for use in many, many tumours. And it has the broad combination therapy potential, which we think is particularly appealing for this therapy. So what is CLEC14A? It's a tumour endothelial marker, which, as I said earlier, it's expressed specifically on tumour vasculature. It has a role in angiogenesis and is expressed in low shear stress, which we see in the poor blood flow in tumours. So you see a disruption of the tumour vasculature, and with that comes a low blood flow, and with that comes upregulation of the CLEC14A. So we see it as a novel vascular disruptive agent. We intend for it to target the CLEC14A expressing cells, and by selectively killing these cells, starve the tumour of oxygen and therefore lead to necrosis in the tumour and inhibition of tumour growth. The CAR-T that we've developed is a third-generation CAR-T. There's a human single-chain variable protein, and that's specific to CLEC14A. There's a transmembrane region, and then there's two co-stimulatory regimes, domains, A and B, and CD3 theta. We're specifically using, because of the risk with vasculature, we decided to move from a viral CAR-T to an mRNA CAR-T to increase the safety profile of the product. This has been used with other groups. Carl June, for instance, published a paper on it 2018 using this approach. So we think it increases the safety profile quite significantly. We've been using it recently over the last couple of years to look at the preclinical models, which I'll go on and explain, and we've been optimizing the manufacturing of it also. So in terms of where do you see CLEC14A? As I said earlier, it's specifically upregulated in tumour vasculature. We've taken a number of microarrays and autopsy material, and we've stained it. And on the left, your left-hand side, we've got healthy tissues, and on the right-hand side, we've got tumour tissue. And as you can see, there's a significant elevation in the tumour tissue of the expression, with high to moderate staining in tumour and low or minimal staining in the healthy tissues. There's a number of tissues which have stained higher, but what we saw was that was very specific to particular vessels within that tumour. And we believe, and subsequent studies have shown us, that we believe that this is due to them being collected under autopsy, where obviously if there's a a delay in getting the tissue taken at autopsy, you will get a reduction in blood flow, and therefore you will see an upregulation in CLEC14A. And our subsequent studies have reinforced that in healthy tissue, you actually see low or minimal staining. So with that in mind, we've chosen our target, we've optimized the target. We then looked at some tumor models, some in vivo tumor models, using 3 in vivo tumor models, the RIPTAG model, the PDAC, and the Lewis lung carcinoma model. And what we saw in all 3 of them was, if you look at the top, I don't know if this is if you look at the top here, you can see that there's a significant reduction in the tumor size. And with that was a reduction in the vasculature of the tumours. So we saw a reduction in the vasculature, and we saw that both in the area of the vessels and in the staining of the vasculature, and this then led to the reduction in the tumour size. So importantly, also, we saw that there was no CAR-T-mediated toxicity in these studies, but I'll go on and talk about some more. So we saw that it actually did what we thought it was going to do, but then we had to show, because it was vasculature, quite clearly there's quite a safety concern there. So we went on to do quite a significant amount of safety studies, and we've actually looked in over 300 mice using this product. So the first one here, we've got some healthy mice, and it shows that the in vivo toxicity is not present at all. What happened? Sorry, I'm using the wrong thing. Here you've got the infusion, the radiation of the mice, and the infusion of the product, which was mice T cells. And then we left them over a 45-day period. And what we saw was the cells continued to persist throughout the experiment. Importantly, the mice showed no change in their normal behaviour. They showed a consistent gain in the body weight. And when the mice were sacrificed and the T cells taken, we saw that there was still an interferon neuron response when they were exposed to either human or mouse recombinant CLEC14A. We then took the histology from these mice, and again, we looked at all the major organs, and we saw no indication of any signs of toxicity, which gave us quite a lot of confidence. These were the first experiments that we did. We then did an additional over 200 studies in other mice, some with actually quite high levels of the product. And again, they've shown no toxicity in any of the studies. And the mice acted normally and gained weight as normal. So I think the safety profile of it is quite encouraging. We then went on and had a look also about whether there was any selective recruitment or expansion of the CAR-Ts, and indeed we didn't see any, either in peripheral blood or the liver or the lung. And because it's a vascular agent, and we were particularly concerned about these patients maybe receiving surgery to resect the tumour, we had to understand whether there was going to be impact on wound healing. So we used a model where we looked at the wound healing capacity in mock-treated animals compared with the CAR-T-treated animals, and we saw no difference. So it would seem that the wound healing capacity is not affected in any way. So in summary, the preclinical summary data that we have today, we actually find really quite exciting and really quite reassuring. We've shown that the CLEC14A expression is particularly high in the tumour vasculature, and it's low or minimal or indeed absent in the normal healthy tissue. We've selected the GMP process. We've got a suitable target, which we've worked hard to make it as specific as possible. And we've also got electroporation conditions, which we're actually incredibly pleased with, given its really high transfection in these mice, in these cells. The affinity of the product has been shown to be dose-dependent, which shows that there's the ability to discriminate between the target tissues, between the tumour, where it's high, and the healthy tissue where there's just normal levels of expression, or low levels of expression. Efficacy has been shown and demonstrated. I think the 50% reduction in tumour size is remarkable. We believe that this can actually be increase still further if we're using this in combination with other products and other therapies. And that could be something as simple as a resection of the tumour. It might be enough to reduce the tumour size to then make these patients be able to undergo surgery, whereas before they would not have been able to do that. Or we could use it in other combinations, other CAR-T combinations or PD-1 inhibitors, or the variety. In the preclinical safety data, in which we've got over 300 mice, it says 200, but we've actually got some more now. We haven't seen any issues at all, which is particularly important in this target. And what we've found is we've gone to the regulators, and we've spent a lot of time going to the regulators and speaking with the KOLs, because we appreciate that there's a nervousness of going for a vascular agent. And based on the data that we've presented to them, the MHRA, which is the regulator in the UK, is particularly happy with the data, and we're going to go back and meet with them again once we've finalised the clinical trial that we're anticipating, to ensure that they're happy with it. But so far, they were incredibly happy with the data and thought that the safety profile of the product was very reassuring. So that's the summary of the product. And what we believe is that it's a novel therapy, it's a novel target, which we think has particularly strong capability to be used in solid tumours. And as I said, that could be used alone, as a standalone therapy, or in combination with other ones. There's no competition. It's a very crowded field in the CAR-T therapy. We don't know of any other company that's looking at this and looking at this particular CLIC14A target. We think it's a very useful target. It can be differentiated into very many different existing cancer therapeutics, and it's very versatile, as I said earlier. There's an improved safety because we've chosen to use the RNA CAR approach. What we have done, and I should have said that earlier, was we've used the gamma retrovirus in all of the animal studies because we thought that was the worst case scenario. Is that, obviously that's going to be a persistent effect, whereas RNA, it's a transient effect. So we deliberately decided to look at the gamma retro transduced product. That does two things. It gives us that confidence that even with persistence, there was still no safety concerns, but it also allows us to, if we wanted to in the future, move to a retroviral or a lentiviral way of producing the product, then we could. And we've also discussed that with the regulators. And we've also got a comprehensive patent portfolio to support this product. So what are the next steps? We're going on with companion biomarkers and further safety studies. One of the safety studies that we're doing, obviously, is we need to compare the mRNA against the gamma retro, so we're currently doing that, as well as some other studies. We're looking at moving to a fully closed CAR-T manufacturing process, and we think we're very close to that. And then it being transferred fully into GMP, and we're currently looking for CMO to do this. The preclinical study is planned to be completed within the next few months. And we're in the midst of finalising the clinical trial design. We're working with key opinion leaders to allow us to do this, and indeed CRUK are particularly expert at this also. And we're going to have a follow-up scientific advice meeting with the MHRA, which we've agreed to present the further data so that they can be happy with the data and the clinical trial that we're going on to do. And then I suppose the next steps are to find a suitable partner for us to actually move the product on to the clinical trial, and that's currently what we're looking to do.